In Chapter 11, we're going to briefly review the organization and structure of DNA in both eukaryotic organisms as well as bacteria and viruses. And we'll start out with the simpler organisms first. What this initial figure that I've got on this first slide is showing you is the hierarchical organization of genetic material in you and other eukaryotes. And what I'd like you to note here is that we've spoken quite a bit about the double helix, which you can see at the top. And then if you look all the way down at the bottom, you will see a replicated chromosome, which we've also spoken of at great length. But what we haven't really discussed is how that double helix is compacted into a chromosome for the purposes of cellular division, whether it's mitosis or meiosis. And we'll start out by briefly reviewing some information about genetic material in viruses. And here are two images illustrating the structure of bacteriophage genetic material. Really, what I want you to get out of this is that very small packages, viral capsids, contain a great deal of genetic material. And I say here DNA, there's a lot of DNA in a small package, okay? Viruses are very efficient in their packaging of genetic material. There's not a lot of empty space within those capsids, and the compaction can be pretty remarkable. So here is a bacteriophage that has been gently broken open and all of the genetic material released. And you can see that this genetic material is quite voluminous. There's quite a bit here that's all been packed into that little viral head in the middle. So the two things I really want you to understand about viral genetic material is that some viruses have DNA as their genetic material, others have RNA. And regardless of which of those they have, that genetic material may be single-stranded or double-stranded. And as you might imagine, we're living in a world where we are thinking constantly about viruses. This affects the biology of the virus, how it interacts with a host, and certainly from a medical perspective would affect how you attempt to combat a particular virus. But we're talking primarily about bacterial and eukaryotic genetic material in this class. So we're going to move on and not talk much more about viruses. Bacterial chromosomes, I'd like you to understand that these are, as we've discussed in previous chapters, primarily comprised of double-stranded DNA. And while they don't have their chromosomes highly compacted like ours, they do compact them somewhat and store them in a particular region, whereas they don't have a nucleus as your cells do. They do have a region called a nucleoid in which the genetic material is generally stored. And there are proteins within these bacteria, these histone-like nucleoid structuring proteins, and these are proteins that have a positive charge, which allows them to bind to the negatively charged backbone of the DNA molecule and fold it back upon itself. And so these proteins allow bacteria to fit a lot of DNA within relatively small cells because it's been compacted. So where we're really going to dive into deeper detail is in the organization and structure of eukaryotic DNA, and we will talk about how this compaction of a vast amount of genetic material is achieved and why it's important and why that compaction is not permanent, primarily because we need to open this DNA up in order to utilize it. So we've spoken briefly in the past about the organization of eukaryotic DNA into something called chromatin. And so we discussed this in the context of meiotic division. So outside of mitosis or meiosis, the DNA exists in this relaxed state of chromatin. And during interphase within the nucleus, the genetic material existing in this state of chromatin is quite dispersed, a very relaxed state. And the chromatin, though, does represent a fairly significant compaction of this genetic material. So let me give you an example. If you were to take all of the genetic material from a single one of your cells and take those strands of DNA and stretch them out, not in the form of a chromosome, but in their most relaxed double helix state, these molecules that fit inside of every one of your cells, actually inside the nucleus of every one of your cells, would stretch roughly 6 feet, okay? So that is an enormous length of genetic material that's crammed inside of every one of your cells. So how is that long strand of double-stranded DNA crammed into each of your nuclei? Through the compaction achieved by organizing the DNA into chromatin. So this results in over a 10,000-fold compression of your DNA. So we're going to work from the most relaxed state, this DNA double helix, to this most compact chromosomal form. And I encourage you to think of your DNA in a way that, I'm going to use a real-world analogy that might help you understand why compaction of the DNA is important and how it's hierarchically structured. So the analogy I use is yarn. So on the far right, you see a ball of yarn that you might purchase at Walmart or some other craft store, and perhaps one of your parents or one of your grandparents is a fan of knitting or crocheting, and so you may be familiar with these balls of yarn. Now, when I was a child, I would raid my grandmother's yarn basket that was full of these things, and my brother and I would frequently throw them at one another, and that resulted in this neatly organized ball of yarn looking more like something in the middle, okay? Which didn't work out well for my grandmother, because when she wanted to knit or crochet, it's much more difficult to utilize that yarn that's in this messy, knotty ball in the middle. So what we've got on the far right is a highly organized and compact form of a material that could be far less organized and far more difficult to utilize and move around, okay? And think of that as the chromosome, okay? When do we have our genetic material in this form of a chromosome? It's when the cells are dividing, and when they are dividing, they need to be able to quickly and easily move individual chromosomes to different regions of a cell. So in anaphase, when those chromosomes are pulled in different directions, okay, it's important that you're pulling a highly compacted, organized thing that's not tangled up with other bits of genetic material. And so existing in this highly compacted form is the most efficient way of achieving that without difficulty. Now, most of the time, when your cells are in interphase, the DNA does not exist in this highly compacted form. It exists in a more relaxed state of chromatin. Now, this relaxed state of chromatin looks pretty disorganized, but you can compare it with what's on the far left, the raw yarn that comprises or is woven together in an organized fashion to create, or the raw fiber to create an organized yarn, a thread, if you will, of yarn. So if you think of the far left raw fiber as the DNA that's organized and twisted into an organized string of chromatin, like the middle, that can itself be even more organized and compacted into a chromosome, you kind of have an idea of the levels of organization and compaction that we see in the genetic material of eukaryotes. So let's dig into the first level of compaction. So taking this raw DNA and compacting it into what we call chromatin. So what exactly is the difference between a double helix of DNA and chromatin? What chromatin represents is the organization or winding of this DNA, this double helix, represented here by these pink lines, around histones. So these histones are these proteins that are specifically intended to aid in the organization of DNA, specifically in compacting it into what we call chromatin. So much as you wouldn't just collect a 100-foot extension cord after a tailgate and just throw it into a giant pile in the back of your car or in your trailer, you would wind it up around a reel. So too do we wind our DNA around these histones. And what you see here is two types of histones, the light blue and the dark blue. These light blue histones function together in groups of 8, so these histone octamers. And what they serve is basically a giant ball that the DNA can be wound around, much as you would wind up an extension cord on a reel. Okay, and it's right in the right in the neighborhood of 150 base pairs of DNA that are wound around a histone octamer. And what you see is we have a chain of multiple histone octamers in series, one after the other, around which 147 base pairs of DNA are wound. In between those histone octamers are a different type of histone, these spacer histones, that keep these other ones being interacting or knocking against one another. So each of these histone octamers plus 147 base pairs of DNA is called a nucleosome. Okay, so in this, we see 2 nucleosomes that are separated from one another by an H1 spacer histone. Okay, so this is this first level of organization in which our DNA is wrapped around these histone octamers. And here you can see a micrograph of DNA with these nucleosomes quite visible. Okay, so you see stretches of DNA that are in between all of these nucleosomes, and these nucleosomes are the little balls, and that's where you've got a histone octamer with 147, so 8 histones and 147 base pairs of DNA wound around them. Now, the level of compaction increases further as these nucleosomes are stacked in concentric circles. Think of a spring, okay? So they're wound around in concentric circles in what we call solenoids. So a solenoid is a ring, and what you don't see here is the ring below and the ring above, but we will see that in a subsequent image here shortly. But you take these nucleosomes and bend the DNA with these nucleosomes in concentric circles, stacking them one upon the other to create these solenoids. And these solenoids are what are circled and wrapped to create more dense chromatin fiber, as you can see above. We'll see that here in a second on the next slide. And then that chromatin is tangled into what we organized, I won't say tangled, into what we call a chromatid. So you see here these sister chromatids of a replicated chromosome. So let's look at the whole picture, somewhat similar to the green one I've shown you before. But starting at the bottom, we have our DNA double helix, okay? And then we have the DNA double helix wound around these histone octamers, with the H1 spacers, okay, to create the nucleosomes. Those nucleosomes are wound into concentric circles. You can see that much better here in these solenoids. And so we have solenoid stacked on top of solenoid, stacked on top of solenoid, okay? And then you see where there are these looped domains. It's this tube or cylinder created by stacked solenoids that is itself wound into circles, creating these looped domains. That is where we get the level of chromatin fiber. And so you can see that chromatin fiber actually represents an extreme compaction of the DNA, because the chromatin fiber represents looped domains of solenoids, which are comprised of nucleosomes. So we've got some hierarchical compaction there that leads to a highly condensed form of this genetic material that we call DNA. Okay, so while this chapter is going to be quite brief for us, this is the first part of what I want you to get from this chapter. I want you to have a solid understanding of the organization of chromosomes in eukaryotes and a basic understanding of viral and bacterial chromosomes based on what I've gone over. Okay. Now, the second portion of chapter 11 that I'm going to cover here is the second listed here, these repetitive regions. So our genetic material can be classified as either unique regions, okay, regions that occur only in one spot of the genome, or sometimes when genes have been duplicated, there might be a couple spots where a gene is present in the genome. These regions we're going to spend a lot of time discussing. So these genes are things that are transcribed and translated. They are used to make specific proteins. We're going to spend the next few chapters digging into genes and how they are important and how they are used. But one thing we're going to touch on at the end of this chapter is repetitive regions. So these are portions of the genome, the other portion of the genome. They're not unique. They might be present in numerous places in the genome, and they don't necessarily have a known or clear function. Okay, and so section 11.5 in your text reviews repetitive regions. Now it goes into a great deal more detail than I am going to go into. What I want you to get out of this is what I'm covering here. I want you to understand what repetitive DNA is, and I want you to understand how it forms. I'm going to talk about that. And then I'm going to give you an example of how it's used, which is something we'll touch on again in subsequent chapters, maybe the end of this week or early next week. Okay? But all I really want you to get out of this is what I'm going to be presenting to you. So first, I'm going to show you an example of repetitive DNA. Here you can see the DNA sequence from 4 different individuals. And what we are looking at is the same portion of the same chromosome from these 4 different individuals. So number 1 might be me, number 2 might be you, and 3 and 4 could be your siblings. Okay? And what you see here is that for our chromosomes, let's just say it's one of our chromosomes, the chromosome we each got from our mother, chromosome number 1, we all got this from our mother, me, you, and your 2 siblings. Okay? So what you see is that in this region, we have the same things to the left and right where it's not shaded. Okay? Our genomes are identical. But what's in between is a region where there are 4 nucleotides repeated, GATA. And some individuals, like I, have this repeated 8 times. You have it repeated 9 times, as does one of your siblings. And your other sibling, participant 4, has it repeated 10 times. So these regions are quite interesting for a number of reasons. The first is how they arise. The second is how can we use these to understand the biology of populations and also use it in forensic settings. Okay, so I want you to think about how do these different versions of this gene region evolve? How do we end up with different numbers of repeats in different individuals? Okay, first I want you to think about how you, number 2, could differ from individual number 4. We're looking at the chromosome your mother gave each of you. Clearly, your mother gave you and number 3 the same chromosome, number 1, but what she gave to your sibling, number 4, is her other chromosome, number 1, right? So your mother would be heterozygous. She has a chromosome with 9 repeats and a chromosome with 10 repeats. She gave you and 3, The one with 9 should give individual 4, the one with 10. Okay, now the way in which these different repeat lengths form is during DNA replication, okay? And that's how most mutations arise within our genome, and they can be passed to our offspring, okay? Now they arise due to mistakes that happen during replication, and we've got a whole chapter on mutations to genetic material, but this is a very particular instance of a genetic mutation. So variation in the number of copies of these repeated sections can arise when a new strand is being made. So here we've got this blue daughter strand that's being synthesized from the left to right, and it is using the green template strand as a template for the synthesis of this new strand. We've already learned about DNA replication and how that works. But what happens is, because the region that's being copied has some repeats of this nucleotide sequence, sometimes the new strand that's being made might lose its association with what's already been copied. It might disconnect from the green strand and fold back upon itself, and replication might start again from a spot that had already been replicated. Okay, so what you can see is there are a couple repeats here that are getting done twice because that little bit looped out. And what that means is the new strand that's made is longer than the original template strand. Okay, and so for the time being, we're going to end up with a chromosome that has a little loop in it. Okay, but in the next round of cellular division, if blue and green, these two blue and green strands separate from one another and each of them gets replicated, okay, we're going to end up with the blue strand ending up being a longer chromosome than the green strand that's replicated. And so over time, we end up with different cell lines that have different lengths of this repeated region, and we end up with different alleles within the population, as we saw in the previous slide. And so we can use this information to compare individuals to see how many repeats of a particular region that is full of repeats individuals have. Okay, and so what we use is a technique called PCR, the polymerase chain reaction. We've not learned about that yet, but we will. And what we do is we take DNA from different individuals and we make lots of copies of their DNA for this particular region that has these repeats. And then we can compare the size of my DNA to yours to your siblings. And the way we do that comparison is comparing the size. And we use a specific machine and setup to separate DNA by size. Okay? And so there's an illustration below that shows the genotypes that are possible for this particular locus. So if there are 3 alleles, allele 1, 2, and 3, you'll note that allele 1 is shorter than allele 2, which is shorter than allele 3. And the length of these is based upon how many repeats they have. So if you are homozygous for allele 1, what that means is, if we look at this chunk of your DNA, okay, that includes this repeated region, both of your chromosomal copies of this repeated region are short, okay? And so you can see down below in that figure, there's a blue bar under the 1-1 that is pretty far down to the bottom of this rectangle. Okay, so the blue bar is lower than the red bar, which is lower than the green bar, right? Now, where they're placed in this will make sense in a moment. Okay, so if you're homozygous for allele 2, then you would see something like the genotype 2-2, homozygous for 3, 3/3. But what's interesting is when we see the 1/2, 1/3, and 2/3. These are individuals that are heterozygous for this locus, And so a 1/2, that individual has allele 1 and allele 2. The next one has allele 1 and allele 3, or allele 2 and allele 3. Now, the placement of these red, blue, and green marks is reflective of their size. And you'll notice that the smallest size, or blue alleles, show up further down, and the larger, greener alleles show up further up in this illustration. Now, the reason this is because we use what we call an agarose gel and a procedure called gel electrophoresis. Okay, so that's a mouthful, but here's what's happening. So you can see in the illustration here, we have a power supply. That's just something you plug into the wall, and it's much like something you would, let's say, jumpstart your car battery with. It's got a positive and a negative side. What we're basically doing is taking a small pool of liquid that is in this chamber in front of the power supply, okay, and we're submerging into that liquid a solid gel, an agarose gel. And what this gel is, think of Jell-O. If you've ever had Jell-O, it's damn near the same stuff, okay? It's a semi-solid, and in that semi-solid are some holes that we call wells, okay? And if you put DNA into these wells and you apply a current that runs across this gel, look at how the power supply is connected to the bath in which the gel is submerged, okay? The negative is on the left, or what we would call the top side of the gel, and the positive is attached to the right or bottom side of the gel. We are putting our DNA sample in the well, which is over near the negative electrode. And what happens is when we apply current to this bath in which the gel is submerged, DNA is negatively charged. So think about the direction that DNA is going to move. It is not going to move towards the negative electrode. It moves towards the positive electrode. Okay, because it's negatively charged, it is attracted to the opposite pole, oppositely charged pole. That's the anode. And so the DNA gets pulled through this gel. And if you look at the bottom portion of this image, what you'll see is a bunch of lines on the gel. Okay, we are able to see the DNA as it moves through this gel because we attach some dye to it. Okay, so as the sample moves through the gel, what you'll notice is that these lines are kind of spaced out. Some of them have moved closer to the anode than others, okay? And this has everything to do with how large the chunks of DNA that are in that sample are, okay? And so small samples move faster than large samples. Why is that true? That's because the matrix of this agarose gel is semi-permeable. Think of it as a sponge with a bunch of holes in it. Some of those holes are large, some of those holes are small. The smaller chunks of DNA can move through that matrix faster than the large chunks. Imagine me sprinting into the woods after a rabbit. And I'm chasing a rabbit and the woods become quite dense. So I'm chasing this rabbit through blackberries and bamboo brambles. And the rabbit can get through that much faster than I can. I'll get through, okay? But that rabbit has moved further and further and further away from me because it's so much smaller than me. It's able to get through small gaps faster than I can find or create large gaps for myself. And so the small fragments move faster than the large ones, and we are then able to compare different samples. If you go back to the previous slide, that's exactly what that slide was showing you. It's showing you the different genotypes and how far the DNA for each of that fragment of DNA for each of those genotypes would move. If you're homozygous for the smallest fragment, it's going to move, that DNA from that sample is going to move really far down the gel. If you're homozygous for the large fragment, it's not going to move very far at all. If you're heterozygous, you're going to have different size chunks in your DNA sample, and they're going to move different distances. So why am I teaching you about this? Well, I wanted you to understand that there are these repetitive elements in the genome, and they are part of the vast majority of your genome that doesn't really seem to do a whole lot. Okay? Your genome contains somewhere in the order of 20,000 genes. Okay? That's a lot of genes in your genome, but you have 3 billion nucleotides. Okay? So only about 2% of your DNA are genes. The rest is other stuff, some of which is important, but some of which doesn't seem to have any function, like these repetitive elements that build up due to mutational errors. But we utilize them in ways that I'll show you on this next slide. So let's first look at this gel image on the bottom right. Okay, don't ignore all the text for a minute. You can pause it and look at this in a little bit. What we see here is a gel that has been run for 5 samples. And you can see the wells are right under the numbers 1, 2, 3, 4, and 5. That is where a DNA sample was loaded. An electrical current was applied to the gel with the positive towards the bottom of your screen and the negative electrode at the top. And so the DNA is moved from the well down towards the bottom of that gel. In well number 2, you can see the DNA has moved further than in well number 1. What that tells you is that the region of the genome that we're looking at for sample number 2 is smaller than it is in sample number 1. Okay? So, the other thing I want to tell you about is what's in the first well, M. And this is where we put our DNA marker, and we call this a ladder. Now you might understand why we call it a ladder based on all the lines that appear here, but what it's really used for is gauging the size. It's a size reference, okay? It's really hard for you to read these numbers because the image is terrible, but the numbers start way down at the bottom at 100, and they work their way up to a couple thousand at the very top. But the first one, let's say it's 100, the second one is not labeled, it's 200, the third one is 300, and so on, okay? So as you move up, the ladder is showing you how big a fragment of these different sizes would be. You'll notice that the ladder has 2 really bright bands. The first one, it should be approximately 500 nucleotides, and the second one, 1,000 nucleotides, okay? So what you're able to do with this ladder is compare all the bands that you got in samples 1 through 5 and get a rough estimate of their size. It's not perfect, but it's a rough estimate. So what is in the ladder sample that we loaded into well M is just a bunch of chunks of DNA that we know precisely their size. And so we know where to expect the different bands, okay? And importantly, or interestingly, those 2 bands that are brighter in the ladder, they're brighter because there's more of that fragment in the sample. So the more there is, the brighter it is. These are labeled with a dye. There's a dye attached to DNA in these samples. And so the more there is of a sample, the brighter it is. So with this, we have a good size reference for all the samples off to the right. Now here, I've posed a question to you that illustrates the utility of looking at these regions of the genome that have these repetitive elements. In this case, these repetitive elements are called microsatellites, and I encourage you to consult the text where it describes to you the structure of microsatellites. And in this, we're going to consider a hypothetical crime scene where DNA was collected, okay? So there were 4 people at an event, one of which died under suspicious circumstances. So individual number 1 is dead. Their DNA was run in well number 1. Individual number 3 is a sibling of individual number 1, as is individual number 4, okay? Individual number 4 is their cousin, okay? So we've got of the 4 people that were there, 1 is dead. 2 of them are relatives, a sibling and a cousin. Okay. The 5th individual was a friend. Okay. Now, in sample well number 2, what we have is DNA that was extracted, and this gene region, or not gene region, this repetitive microsatellite region was amplified and loaded into this well from a sample taken under the victim's fingernail. Okay? So if you look at this gel, you can answer these 4 questions. You can tell me which of the 3 people, individual 3, 4, or 5, matches the sample taken from the victim's fingernail. Who has the same genotype as the sample taken from the fingernail, number 2? You should also be able to tell me why individuals 3 and 4 have 2 bands rather than 1. Okay? Why is one of those bands that they have the same as the band that we see in the first lane, or sample number 1? And I also am curious as to why the investigators also have sample 1 in here. Okay, they got sample number 2. This is the skin under the DNA sample under the victim's fingernail. Why do they also include the victim's DNA in well number 1 in order to run this test? Why do they want the victim's DNA in there when they're comparing the crime scene sample number 2 to all these potential suspects? So this is our conclusion of Chapter 11. I hope you find this to be an interesting example, and I'd be happy to chat about this one during any of our Zoom office hours or chats.